I'd like to thank you for joining us and welcome to the webinar. My name is Steve Feeney and I'm an application scientist here at Cellular Dynamics. And today we'll be talking about the ICL Cardiomyocytes and go into some of their general characterization and then into some of the utility and different ways that they've been used out in the field. So before we begin, a few technical pieces. If you're new to WebEx and you haven't done it before, you'll notice up on the top it should say that you're viewing my desktop. If you scroll your mouse over top of it, you'll see a Q&A, or you'll see a bar drop down, and you should have the Q&A option. As we've got a lot of data to get through today, I'll try to answer as many questions at the end as I can. If you don't see the Q&A option, it should be right to the right of it, the down arrow. If you click on the down arrow, the Q&A should be there. And if you click Q&A, it should pop up with a nice dialog box, which you can just type in your questions, and at the end again. We'll try to get to as many as we can, time permitting. So before we get into the cardiomyocytes, we'll start with a little bit of an overview of CDI. The Cellular Dynamics is the world's largest producer of human IPS cells and IPS cell-derived cell types. We're headquartered here in Madison, Wisconsin, and we currently employ over 115 total staff, with about 80 of those being scientists. And we like to say in a field that's only 14 years old that we have over 400 years of cumulative human stem cell experience. In addition to the stem cell experience and scientific expertise you find at CDI, we also have over 460 patents to enable the full freedom to operate, just so you know that when you get our product and you begin working with it, you have all the legal rights to do so. And at CDI, we like to view ourselves as having really four main core competencies. The first is the creation and culture of human IPS cells. The second is genetic engineering of IPS cells. The third is the development of new differentiation protocols. And the fourth is the manufacture of human IPS cell-derived cell types in industrial quantities with scalable production. Also down here on the right, I'd like to point out that in 2011, we were the gold winner for technology innovations from the Wall Street Journal. So a little view of some of the products that we currently offer. Our first product was the ICL Cardiomyocytes, which launched back in the end of 2009, the beginning of 2010. In addition to those, we launched in Q3 of 2011 the endothelial cells, the neurons in Q4 of 2011, and the iCell hepatocytes, which are currently in a pre-commercial release, which means that they're currently a live cell product, but we have been shipping those to many customers. In addition to our inventory of iCell products, we also have a custom MyCell service. So this service offers the individual investigator CDI's expertise in IPS cell reprogramming, genetic engineering, and cell differentiation to really bring the powers and the core competencies of CDI into any individual lab to look at your target patient and in your target cell type. A little layout of how the R&D structure at CDI is organized. As we said in a few slides ago, we have about 80 or so scientists, and of them we have a dedicated production staff, as well as a dedicated QA and QC staff, and that's really to ensure that the product you get is made exactly the same every time to the highest quality standard. We also have an advanced research group which looks into more advanced technologies in the IPS field, but the bulk of our scientists lie in our R&D cell biology and process sciences. And this is where the products that we develop come from. We have the R&D cell biology group to determine the new differentiation protocols, as well as the process science to ensure that the protocols are scalable and reproducible before they make it into the production queue. In addition to the scientific staff, we have a fully functional commercial team. This consists of the product managers who act as the voice of the customers to relay your concerns and your wants back into the R&D organization to ensure that it's incorporated into the product, as well as a field-based global account team and a fully operational tech support and application support team. This is the team I'm currently a member of. We have a U.S. and European presence, and we have a state-of-the-art application and training laboratory. We generate a lot of the user's guides, the application notes, the training videos. And we also offer the certification programs, which are programs which we can provide internally to customers to allow them to come to CDI, see the cells firsthand, and get hands-on experience thawing, dissociating, and working with the cells, as well as tips and tricks on how to use them on highlighted applications. In addition to our commercial team, we also have IPS Academia of Japan, who distributes and supports the iCell products in Japan and the Asian region. So a little look at our manufacturing process. We like to blend a little bit of the ISO and GMP processes to ensure the highest quality. We are not individually one or the other. However, we try to take the best of both to ensure a high-quality product that meets the demands and needs of the customers. Using our patented technology and selection technology, we're able to reprogram the IPS cells, engineer them, differentiate them into the different cell types, and then purify them. This allows us to produce industrial quantities of highly pure IPS cells and differentiated cells. As a little benchmark of what our capabilities are, on a cells per day basis, we can generally grow over 2 billion IPS cells, over 1 billion cardiomyocytes, 1 billion neurons, 500 million endothelial cells, and roughly 500 million hepatocytes as well. The processes that we use are very translatable, scalable, and, as I said, IP protected. So what this means for the general IPS technology. As you can imagine, there are roughly 6 to 7 billion individuals in the world today. And with the IPS cell technology, you can literally take a sample from any of them and reprogram them into the cell type that you want. There are over 200 cell types in the body. So as you can imagine, it's a fairly unlimited product portfolio. And that brings us into the iCell Cardiomyocytes. So the iCell Cardiomyocytes, first and foremost, human IPS cell-derived cardiomyocytes. You can see here in the video on the bottom left, cells beating spontaneously in culture, which you can see just under a normal microscope if you'd like. The cells are typically high 90% purity, usually in the 99% pure. They're cryopreserved and they're ready to use. And that's very important if you're working with IPS-derived cell types, in that it allows you to plan your experiments and use the cells around your experiment design instead of having to plan your experiments around the cells. They're available in virtually unlimited quantities, so as many cells as you need, we can provide you. And in addition to just the cryopreserved cells, which you see in the vial here, we also provide the media as well as the protocols. So for many of the different applications I'll show today, we have a number of protocols that are worked out as well as publications where the cells are being used. And you can feel free to contact me or the technical support group, and we'll provide you with the protocols as best we can. The cells themselves demonstrate normal human cardiac biology, electrophysiology, and the toxicity responses. And they're useful, as you'll see today, across a wide variety of platforms, which make them ideal for discovery and different preclinical drug development. So going into the iCell cardiomyocyte characterization, we'll start with the genomic and transcript characterization. So on the left-hand side, what I have is a transcriptome analysis. This was performed by Roche and is published in Babieres et al., where they took our cells, starting at the IPS cells, through the differentiation, and then with additional time in maintenance and culture. And what you can see here is at the beginning, the stem cell genes, which were categorized by Roche algorithms, show that essentially all the cells were stem cells. The transition genes were off, as well as the cardiomyocyte genes. And through the differentiation process, you see the stem cell genes decline, the cardiomyocytes genes turn on, and the transition genes turn on temporarily and then shut off again. And I should point out here that at day 28 is where we typically cryopreserve the iCell cardiomyocytes. So for the sake of this experiment, Roche took our cells and they continued culturing them for an additional 3 months, so up to day 120. And what they saw was even more important was that the gene expression stayed very stable over time. Stem cell genes remained off, the transition genes remained off, and the cardiomyocyte genes remained on. So we have a very stable cardiac gene expression. Next, they took ambient RNA expression, and they rank ordered 250 different genes from highest to lowest, and they compared our cells at day 28 and day 120 to the ambient RNA profile. And what you can see is there's a very good correlation between these 250 genes and the ambient RNA. And even more impressive is that day 28 and day 120, when they ran the experiments, you see the gene expression is virtually overlapping, showing that these cells do not change too much once they're in culture, which is a great advantage when you're looking at different time courses in your experiments. They continued and they did this with the primary cardiomyocytes as well, cells they received from PermoCell and Celprogen. They did the same analysis and compared it, and as you can see, compared to the ambient RNA, they had not as strong of a correlation. This led the investigator who was doing the work to kind of jokingly say that our cells were more human than human. Simply put, that the ICL cardiomyocytes and adult mRNA show very similar cardiac expression profiles, more similar than primary cardiomyocytes, which have been put into culture. Jumping from the genomic expression into the protein expression, as you'd expect, we've got all the major cardiac contractile proteins that you'd expect. Here you can see on panel B, the cardiac troponin T in a little bit of a wide-out zoom, showing all of the cells are positive for cardiac troponin T and are cardiac cells. In panel C, looking at alpha-actinin, you can see the nice striation in the cells as well. Here in the green, showing the striation in the sarcomere. When you look at panel A, looking at cardiac troponin T and alpha-actinin in the whole population, we have a very high 97.42% by flow. Looking down now at panel D, we have myosin light chain 2V in red and cardiac troponin T in green. And you see that we have a mixed population of ventricular-like and atrial and nodal-like cells, with the ventricular-like cells showing up in the 2V staining here. And next, for cell-to-cell communication in panel E, we've got the connexin 43 staining in red, showing up around the cell-to-cell contacts, showing that our cells indeed are communicating as cardiomyocytes would. Going next into the electrophysiological characterization from the protein characterization, you can see that we have all the major ion currents. This data all comes from Ma et al., which was published in 2011. You can see we have the expected depolarizing currents in the sodium and L-type calcium, the repolarizing currents ITO and IKR, as well as the pacemaker current I-funny, and the maximum diastolic potential setting current of IK1. Now, as we saw in some of the protein staining, we do have a mixed population of different cardiac subtypes, and we saw the same thing when we did the electrophysiological characterization. What we saw were the ventricular cells made up approximately half of the population, with the remaining cells being split between the atrial and nodal-like. This work is all published in Ma et al., and you can actually see the protein expression in another publication by Li et al., which correlates very nicely, showing again about roughly half the cells as ventricular-like. For the electrophysiological characterization, the way we determined ventricular-like was by primarily looking at the ratio of the APD 30 to 40 and 70 to 80, just to characterize whether or not there was a plateau in the cells. So in addition to the normal EPHIS, we also looked at the different receptor, GPCR receptors, where we saw isoproterenol, phenylephrine, and carbachol. So with isoproterenol and phenylephrine, looking at our adrenergic stimulation, we did see an increase in the beat rate. And with the carbachol, looking at muscarinic receptor activity, we saw the expected decrease in beat rate. Lastly, I'll go over the metabolic characterization. This work here on the top left-hand side was performed by Lilly using the Seahorse Biosciences instrument. And it simply demonstrates that the ICL cardiomyocytes do, in fact, use, as you'd expect, the mitochondria for their energy production. Now, following that up, Rana et al. At Pfizer took a look at the shift in energy metabolism when cultured under different conditions at different time points. And they showed that depending on the energy substrate of the media, you can really shift the energetic profile of these cells. With glucose, you'll see they'll be more glycolytic, and as you take the glucose away, they become increasingly more dependent on mitochondrial respiration. These cells actually make an excellent model for mitochondrial respiration due to their heavy oxygen-consuming nature of the cardiomyocytes. Additional work was done by Rachel Legman at Seahorse Biosciences and presented in a poster at Experimental Biology, in which they took our cells and injected fatty acids. And you can see here in pink that after the injection of the fatty acids, the oxygen consumption rate increased as linked by beta oxidation, which they showed here at C, where they injected a compound to block the beta oxidation specifically. Now, when they incubated the cells in an antioxidant, they showed that they were able to actually increase the oxygen consumption with fatty acids. And again, that was blocked, once again, with the compound to block beta oxidation. So with this typical characterization that you'd expect from a human cardiomyocyte, it really opens up a lot of potential assay endpoints in which the cells are being used. You have your classical ion channel and action potential. You have your mechanical activity, your mitochondrial function, cytotoxicity, GPCR modulation, proarrhythmia, and really one big advantage of the iCell cardiomyocytes is that it's platform agnostic, meaning that you can use it across a wide variety of platforms. So you have all the different functionalities. And down here on the bottom, we have just a sampling of some of the different vendors who are currently using our iCell products on their instruments. With all of this utility and broad array of applications, we've seen a large number of publications on the iCell cardiomyocyte. Here's a listing of just some of them, showing a wide utility looking at different gene expressions, the calcium dynamics, different mechanisms of cytotoxicity and metabolism. A wide variety of just for a product that hasn't been on the market for very long, that's growing very quickly. In addition to the publications, I should point out that the cells have also been used for data for two separate IND filings by two separate companies to date. So going more into the utility and the different areas where these cells are being used, the first thing that everybody thinks of for the cardiomyocytes is looking at the electrophysiology. Starting here with the manual patch clamp, looking at the sodium channel current. This is published again in Ma et al. You can see normal sodium current. This is a little slow as it was collected at room temperature, and the inactivation and deactivation curves that you'd expect. Going more into the pharmacology space, we're looking at IKR block. We saw the expected prolongation of the action potential, as well as the generation of early after depolarizations. So a very big red flag when you're looking at arrhythmia, and it's all at the clinically relevant concentrations of E4031. The next usage here in automated patch clamp was published and done by Nanion, looking at the sodium current with TTX block, as well as the action potential with TTX block again. And it's important to look at these cells when you compare them to a normal overexpressed, say, HEC line or CHO line, where you're really just looking at one individual ion channel. You can use the iCell cardiomyocytes and look at a specific ion channel in the individual channel recordings, or you can look at the action potential and see the interplay of all the major ion channels. It offers one model, or one cell type as one model for all the different ion channels, instead of trying to translate your data across different cell models. Next, we move on to the MEA, which looks at a population of cells and measures the field potential instead of the membrane voltage. So you can see here, we're looking at L-type calcium block with nifedipine. We get the expected shortening of the field potential duration, as the calcium is primarily responsible for the plateau phase. So blocking it, we'll see a shortening in the cells, which is quantified down here below, and a shortening of the field potential duration, a shrinking of the calcium wave, and an increase in the beat rate. Now, this MEA work was performed by Roche and is currently published in Gu et al. And what they did was, from the MEA work, they continued the work, and they looked at the Excelligence from ASEA. Now, Excelligence measures impedance, which is more closely related to the physical contraction of the cells, as opposed to the MEA, which looks at the electrical activity of the cells. Now, this is best exemplified by blebbistatin, which is an excitation contraction decoupler. So what you see is, in the Excelligence system, you see a strong shrinking of the signal here, of the amplitude of the contraction strength when blebbistatin is applied, Whereas if you were to look at this on the MEA, you really wouldn't see much of an effect because you're measuring the electrical activity, which is largely unaffected. They took this and they looked at a number of different compounds. Here we have tetrodotoxin for sodium block, ZD7288 funny current, so looking at your pacemaker activity. Again, we have our GPCR modulation and our calcium block. And they took this data and they compared a number of known arrhythmogenic compounds. So here we see the nice, stable, clean beating in the DMSO or aspirin-negative controls, as well as a sample of what the beating looked like with the application of different compounds. From this data, they were able to generate a predicted pro-arrhythmic score. You can refer to the paper in toxicological sciences to take a look at how they calculated the predicted pro-arrhythmic score. However, when they rank ordered the ICL cardiomyocytes from what it predicted to be the highest risk to the lowest risk, what they found was very interesting. So when they compared them to their standard HERG and QT tests, which didn't pick up a couple compounds like wabin or Aquino-19, and they compared that to the animal in vivo studies, which did pick up a liability, they found that the ICL cardiomyocytes picked up the liability as well. And when they compared the compounds which ranked low by the ICL cardiomyocytes, but were detected by the HERG and QT, but not the in vivo animal models, they found that the ICL cardiomyocytes, again, represented the in vivo phenotype over their other models. And for their standard compounds, which were detected in everything, they were detected as well in the ICL cardiomyocytes, and the compounds which were detected in nothing as no risk were, again, ranked very low with the ICL cardiomyocytes. So here you have a good example of the ICL cardiomyocytes really being used as a more predictive model over many other in vitro tests to look at the in vivo phenotype. In addition to the electrophysiological activity, you can also look at contraction. So this is adapted from Papella et al. And some work that was done over at Pfizer, looking at the cells in the ion optics. So the ion optics takes calcium fluorescent measurements as well as edge detection for the contraction strength. And when we block the calcium with nifedipine, so an L-type calcium blocker, you see the dose-dependent decrease here in the top left corner. But you also see the decrease in contraction, as calcium is very important for cardiomyocyte contraction. And so when you add compounds that increase or decrease the contraction strength or the calcium, you see that as well on the ion optics, which shows the ICL cardiomyocytes have the potential to be a good positive or negative inotropy model, looking at contraction strength, which is something that you wouldn't find looking at just looking at normal ion channel activity. Sticking with the calcium fluorescence, some recently published data as well on the flipper tetra system by molecular devices, Sorenko et al., looking at the ICL cardiomyocytes for chronotropy measurements. So looking at the adrenergic isoproterenol and epinephrine increasing the beat rates, as well as all the compounds expected to decrease the beat rates, decreasing the beat rates. In addition, the flipper calcium transients could also be used to reflect the electrical activity on the membrane. So here we have action potential prolongation with cisapride showing up as well on the calcium transients. And this can all be quantified using the Peaks Pro software, which you can purchase with the flipper tetra system. And more importantly, the flipper tetra really kind of brings out more into the higher throughput screens. So you can perform these cells at 96 well plates or 384 well plates, which make them very conducive to high throughput screening and discovery experiments. So sticking with that, this work was done on the Image Express microreader, looking at a JC10 readout using Annamycin A as a mitochondrial toxicant. With the ICL cardiomyocytes, you can see here in orange the healthy mitochondria before and then after the application. With the high throughput Image Express, you can measure all of your wells, you can analyze the data and quantitate, and really start to dive into more of the advanced biology of the ICL cardiomyocytes. Now this can be used for general cell toxicity. So here we have just straightforward cell titer glow data from Promega, taking a look at known toxicants, and they all show their expected cardiotoxicity. However, it's important to point out that really any out-of-the-box kit that you can use with other cell types, you can also use with the ICL Cardiomyocytes. So as an example, we have the GSH glow to look at oxidative stress, cash-based glow to look at apoptotic activity, proteasome glow and HDAC glow to take a look at the histone deacetylase as well. And you can really take any of these specific endpoints and find out not just whether or not your compound kills the heart cell, but you can start to look at why it kills it. And this was something that was done very well by Jennifer Cohen in a 2011 publication where they looked at sunitinib toxicity. So the sunitinib toxicity had a well-known kinase pathways that it was influencing in rat models that were being used. However, when they applied the cells to the ICL Cardiomyocytes, they found new mechanisms of toxicity. They measured a number of different endpoints. Here we have on this graph just the ATP, the LDH, caspase, and GSH, as well as a number of the kinase pathways which were activated and inactivated, and showing that a different set of kinases in the human model were being activated when compared to the rodent model. Just showing that when you're doing your toxicity, it's important to have a physiologically relevant model to be studying what it is you want to be studying instead of a different phenotype. And then once again here in the cell type specificity, on the right-hand side, we have a comparison of ICL Cardiomyocytes to K562, so a cancer line. Cancer lines are very common in pharma and drug discovery, but they're not exactly representative of normal physiology. Here we have SAHA, a cancer drug being applied to the ICL Cardiomyocyte, as well as the K562s, and you see an increase in apoptotic activity and an increase in and a decrease in the viability, increase in the cytotoxicity in the cancer line, as you'd expect from a cancer-specific drug. But you don't see the same effect in the ICL Cardiomyocyte. So once again, it's very important to use a very relevant model to study what it is you truly want to study. And with the ICL Cardiomyocytes, you get all the different pathways biology that you'd be looking for. So in addition to these specific endpoints, the ICL Cardiomyocytes can also be transfected efficiently. As an example here, I've got the ICL Cardiomyocytes with their normal RFP in red here, and the transiently transfected GFP showing up here in green, just to demonstrate that the cardiomyocytes are transfecting. And as you can see from these flow plots, we've got very good efficiencies at 42 and 34%. However, we also did this at day 4 and day 12. We got very similar transfection efficiencies, which really makes for a very convenient workflow when you're using the cells. In addition to being able to transfect these at different times, the vectors also can maintain activity over time. So a lot of times when you're working with a heavily proliferating cell line, your vector can be kicked out. However, the cardiomyocytes don't proliferate. So once the vector's inside, oftentimes it can stick around. And here we show this with a pre-luciferase reporter vector that was transfected at day 4 and induced on day 5, and transfected at day 12, induced on day 13, and then an additional one that was transfected at day 4 and induced on day 13. And we see that we've got a very similar response to isoproterenol throughout each of the different time points. This really opens the ICL Cardiomyocytes to a wide variety of investigations, and you can transfect in a whole number of different plasmids. Here, just as an example, we have the cyclic AMP reporter element again. We've got the antioxidant response element and the NFET response element as well, looking at luciferase activity through induction with different compounds. So you can really take this and look at the pathways you want to look at in the model that you want to look at them in. And just as a further example, we've got the antioxidant response, looking at the cytotoxicity with TBHQ, showing an increase in the antioxidant response in black and a decrease in viability in red. And then taking that and blocking it with ascorbic acid, so an antioxidant, to suppress the antioxidant response element. And you can see, again, the expected decrease in the antioxidant response luciferase. In addition to just plasmid transfections, you can also do RNAi. This is some work that was performed with Vitrogen using lipofectamine RNAi max, looking at just a toxic small interfering RNA chain. Here we have the non-transfected cells, as as well as the transfected cells here on the bottom, just showing that without the transfection reagent, we maintain good viability. However, once we transfect a toxic RNAi chain, we see the decrease in viability as you'd expect. So this really opens up a whole slew of investigations. You can look at different, you can activate and inactivate different genes and different regulation pathways. And you can start really using the ICL cardiomyocytes as a model for different cardiac diseases. And one such disease which has been recently published using the ICL cardiomyocytes is cardiac hypertrophy, which is simply an increase in the heart mass due to an increase in the size of the cardiomyocytes. So as you can see, essentially the small heart becomes bigger through a large number of pathways, and there's different ways to read this, such as the cell size, an increase in protein synthesis, an increase in different fetal gene expressions. And one that was performed recently by the Medical College of Wisconsin, Zia et al., was looking at the ICL cardiomyocytes increase in size when stimulated by isoproterenol, a known hypertrophic reagent. And what they found was when they stimulated them, they did see a statistically significant increase in the size of the cells compared to the unstimulated condition. Now this becomes even more important when you think of the implications of IPS technology. So this work was done with an NHLBI grant, taking a look at 250 patient samples from a hypergene cohort. So this is a large group of genome-wide association studies looking for left ventricular hypertrophy in hypertensive patients. And left ventricular hypertrophy is a very common marker of potential cardiac failure and cardiac disease. And what CDI is doing with the Medical College is we are deriving IPS cells and cardiomyocytes from all 250 individuals, and which will provide the cells to the Medical College so they can induce the hypertrophic phenotype and perform molecular analysis to really use this clinical population in this genome study, take a look at the genomic data compared to the in vivo or in vitro phenotypic data, and see if they compare and if they can find known pathways or find new pathways and interventions for cardiac hypertrophy. Now, internally, looking at hypertrophy, one thing that we've done a lot of work internally on is looking at endothelin and BMP expression. So again, endothelin-1 can induce cardiac hypertrophy, and BMP expression is one of the markers of cardiac hypertrophy. And when applied to our cells, we see the expected increase in BMP expression by qPCR. But what's even more important is that when we compare this across lots, we saw, as you can see here, a very similar EC50 of the endothelin across multiple lots, showing a very consistent and well-performing assay, as well as a very consistent product. And in keeping with the platform agnostic aspect of the iCell cardiomyocytes, we also looked at endothelin induction of BMP in high-content imaging, flow, and ELISA. And you can see here in high-content imaging before and after endothelin treatment, you've got a very low concentration of BMP to a very high one, the same with flow. But what's even more important is the EC50s between each assay are very similar between each other. Just once again demonstrating that the iCell cardiomyocytes offer a very powerful tool to look at a number of different pathways in a physiologically relevant model in a number of different ways to really suit your needs. So, in summary, the iCell cardiomyocytes are an excellent tool for recapitulating in vivo human cardiac myocyte biology. They provide an ideal tool for interrogating a cell model that is relevant to what you're looking for. They're platform independent. As you saw today, there is a wide number of platforms and uses that these have been used for, as well as an ever-expanding list of publications. And they're available on demand. We have them in inventory, so when you need them, we can send them out. So where can the iCell cardiomyocytes be used in the drug discovery pipeline? As I'm sure you're well aware, the drug development is a very expensive process. And really, the big motto is to fail faster. The sooner you can bring in a relevant model in a high throughput manner, the better. And the iCell cardiomyocytes are human origin and have very good functional relevance, and are amenable to many high throughput modes of analysis. So it really brings them more into the drug discovery earlier on in the process to get a good arrhythmogenic pathway biology mechanisms. When you go into the toxicology, their large quantity and purity offer an excellent model so that you know when you're killing the cells that you're killing the cardiomyocyte and not a fibroblast or a contaminating cell population. It lets you know that the targets you're looking at are the targets you want to look at. And because they're IPS, it offers a whole new realm in clinical testing, in that if you have an outlier patient who expresses toxicity, you can always take a sample of the patient, and using the MyCell services, we can generate IPS cells as well as the different cell types which you wish to investigate. So for the purposes of cardiac biology, we can generate the cardiomyocytes from them. And you can take a look at that phenotype earlier to ensure that when you get back to clinical testing again, you don't have the same kind of problems crop up repeatedly from a small genetic population. So with that, I'd like to thank you for joining our webinar. If you have any questions, please feel free to contact app support. You can do so through our website, www.cellulardynamics.com, through our email, just support@cellulardynamics.com, or over the phone, 877-310-6688. And at this time, I'd like to open it up to any questions that may have occurred during the webinar. So our first question here is, how many cells are in a unit? So a single unit contains 1.5 million platable cardiomyocytes. And what I mean by platable is that we understand that when you add cells to a well, not all the cells you put in there will attach. And that factor is included in the C of A of every lot that we produce and is tested by our QC. So we overfill the vials to ensure that at least 1.5 million cells will attach to your plate when you receive them. And this generally comes down to enough cells to confluently plate a 96-well plate per unit. The next question is, how long can they be maintained in culture? So we tend to say that the cells can be maintained indefinitely in culture. They're non-proliferating, which means as long as you continually feed the cells, you do not need to passage them, and they just continue to beat and stay in culture. You saw in the genomics data where they kept them out for an additional 3 months. Internally, the longest we've gone is a year, at which point we just harvested the cells because we wanted to take a look at their protein and purity levels. Next is, what shape are the cells? The cells tend to take on a fairly amorphous shape in culture. This differs a bit from a primary cardiomyocyte when you first dissociate it from a cell. So if you've worked with primary cardiomyocytes, they tend to be very rod-like in structure or elongated. However, the ICL cardiomyocytes, most likely because they haven't been in a structural organ and because they're growing on on a flat, hard tissue cultured plastic surface, tend to be very amorphous, rounded, and shapeless when they're plated at a low single cell density. And then the last question I have is, does the RFP interfere with the assay readouts? So with the transfection slide I showed, the RFP showing up, and that's stably expressed in the ICL cardiomyocytes. However, it can increase the background in some assays. Typically, it's a very low fluorescence, so a lot of assays do often work around it if they're reading in the red channel. However, we also offer our RFP-free cells in addition to the RFP cells. So if you're concerned about this, you can contact your account manager, and they can provide you with the RFP-free cells, which are developed from the exact same donor source and the exact same line. The only difference is one line had the RFP engineered in, the other line didn't, and then they're generated the same exact way. And with that, that takes up all of our time for this webinar, and I'd like to thank you for joining us. And if you have any questions, which I wasn't able to address in the time that we had today, I will be sure to get back to you through email after the event. Thank you.